Okay, we're all set to go. Go ahead, Anna.
Okay, hi everybody. Welcome. We are really excited to have you here with us tonight. Hola a todos, bienvenida. Estamos muy emocionados de tenerlos aquí esta noche. You are joining the Hope, Empower, and Rise, or Her Path to Science webinar series, which is part of our Seeds of Success program, which is open to 6th, 7th, and 8th grade students here in the New Haven Public Schools. Se están uniendo a la serie de seminarios web Hope, Empower, and Rise, que es parte del programa Seeds of Success, abierto a estudiantes de sexto, séptimo y octavo grado en las escuelas públicas de New Haven. Whether you're joining us on YouTube or on Zoom, we're really glad to have you here. We're going to have a great conversation tonight. Ya sea que nos están viendo por YouTube o aquí mismo en Zoom, nos alegra mucho que estén con nosotros. Vamos a tener una muy buena conversación esta noche. Before we get started, just a couple of things. Tonight, our speaker can answer your questions in English or Spanish. Esta noche nuestra invitada puede responder sus preguntas en inglés o en español. So feel free to ask in either language. No duden en preguntar en cualquier idioma. If you're watching on YouTube, you can drop your questions in the YouTube chat and we're going to read them for you. Si nos están viendo en YouTube, pueden soltar las preguntas en el chat de YouTube y las vamos a leer por ustedes. And if you're here on Zoom, you can ask your question live or type it in the Zoom chat and we'll ask it on your behalf. So, si están aquí en Zoom, pueden hacer su pregunta en vivo o escribirla en el chat de Zoom y nosotros la haremos de su parte. Así que no tengan miedo de hacer preguntas. Don't be afraid to ask questions. I'm going to now introduce tonight's special guest, Dr. Janitza Montalvo Ortiz, which is a scientist at the Yale School of Medicine, where she is an assistant professor of psychiatry. She studies how our genes and our experiences, like stress or trauma, can affect our brain and our mental health. She grew up in Puerto Rico, earned her degree in biology from the University of Puerto Rico, and went to get her PhD in neuroscience from Northwestern University. Now she runs her own lab right here at Yale. She is a brilliant scientist, a role model, and we are so lucky to have her with us tonight. So please join me in welcoming her. Les voy a presentar a nuestra invitada especial de esta noche, la doctora Janitza Montalvo Ortiz, la cual es científica en la Escuela de Medicina de Yale, donde es profesora asistente de psiquiatría. Estudia cómo nuestros genes y nuestras experiencias, como el estrés o el trauma, pueden afectar nuestro cerebro y nuestra salud mental. Creció en Puerto Rico, obtuvo su bachillerato en biología en la Universidad de Puerto Rico y luego el doctorado en neurociencia en la Universidad de Northwestern. Ahora dirige su propio laboratorio de investigación en Yale. Es una científica brillante, modelo a seguir y tenemos mucha, mucha suerte de tenerla con nosotros esta noche. Así que acompáñenme a darle la bienvenida. Go ahead.
Thank you so much. Such a nice introduction. Muchas gracias. I'm gonna start to share my screen, so stay with me while I do that. Okay. I think it should be all. Is it working? Not yet. I think I did that. Yes. Desktop one. Let me do that again. Okay. Yes. Good. Okay, perfect. So, first of all, thank you so much for the invitation to talk to Seeds of Success program. I'm very excited to be here tonight, and I'll be talking first about the science I do, and then we'll end up with a brief journey on how my career path has looked like. And for that, I wanted to start with what it made me a scientist, and what kind of curiosity drove the science that I do today. So since I was a kid, I've been always curious about, what makes us us? How is our identity defined? What shapes who we are? And there are two beautiful mysteries behind it. And I would say our brain and our DNA. So for that, I wanted to introduce myself. I'm the youngest of four girls, and I want to say that each of us have totally different personalities, very unique preferences, but yet with always something that makes us similar, makes us sisters. And if you look closely, you'll also notice that we have different eye color, we have different hair color, slightly different skin color, but yet with the same smile. And my family, being from Puerto Rico, is very similar to any other Latin American family, Hispanic family, and like the one we see in this picture, which is from a Disney movie. Does anyone recognize which Disney movie this is?
It's Encanto.
Encanto, right? This is one of my favorite movies, and this nicely depicts the Madrigal family. And as we know, each of the characters in this family has very unique personalities, likes, and very special gifts that makes them special, and also very unique but yet blended physical traits. And we see some of these physical traits are passed down through generations. You know, our eye colors could be similar to our grandparent, maternal grandmother, or great grandparent. So these are passed down through generations. We know that now that through genes. So what is a gene? A gene is a fragment or sequence of the DNA that acts as an instruction manual to build stuff, that tells cells how to build brown eyes or make insulin. And how do we know the fundamentals of the gene functions? Is through the Human Genome Project. This was completed more than 20 years ago, where scientists read all the DNA sequence and mapped the now over 20,000 genes that we have. But this was just the beginning. This discovery basically paved the way for a genome revolution, welcoming us to the post-genomic era. What does that mean? It led to discovery of hundreds of thousands of what is called genetic variants that are now associated with multiple variety of diseases and disorders. Here I'm showing a map of the GWAS catalog, like genomic catalog. And each of these lines are the chromosomes. And the zoom in is basically each dot refers to a genetic variant that has been identified as associated with a disease, Alzheimer's, Huntington's, alcoholism. And this is all deposited for public access in a catalog that could be accessed through the internet. In psychiatry as a complex trait, we know now that based on the study of 2014, we're able to start identifying not a single gene because it's a complex trait, but multiple genes that are known to contribute to the risk of developing schizophrenia later on. About several years after that first study, we started to study multiple populations, not from a single background, genetic background or ancestral background, but also European, East Asian. We started seeing this boost in gene discovery. And we realized more than ever that for complex traits or disorders like psychiatry, you know, it's not a single gene that makes you have the disease, it's multiple genes. And we see this across the board for different psychiatric disorders, having disorders like depression, bipolar disorder, and schizophrenia as one of the most successful cases of identifying genetic variants of risk. However, there is a lot of work to be done for other psychiatric traits that are less known, like addiction, like disorders, like cannabis use disorder, opioid use disorder, and also other psychiatric traits like suicide, anorexia nervosa, autism, and so on. And now we recognize there has been hundreds of genetic loci identified. We'll now learn that they're highly polygenic, which means, as I told you, that there's not a single gene disease. These are multiple genes disease. And also, we discover there is a highly significant heritability, which means that the risk to develop the disorder are coming from the genes that we inherited from our ancestors. And even with all these successes in the field, there's a lot of work to be done. Even with all the hundreds of genetic variants identified, there's still a small portion of the risk that is explained, leaving a high portion of the heritability called as missing heritability because we don't know where the risk is coming from. We also know that the functionality, what these variants, these genes do, over the 90% of these is still unknown. So we need to do a lot more work to understand what their function are in our body. And also we know that the DNA sequence alone cannot capture environmental effects. So we are not necessarily defined 100% by our genes, but also about things that we eat, how much exercise we do, things that we learn, and so on. And also, I would say that a huge gap in the field is also the lack of diversity of the populations that are studied in these genomic studies. And this is highly problematic. Just to give you an idea, this is comparing by the global population proportion. The red one is constitutes over 90% of genomic studies that are being disproportionately represented by individuals of European ancestry, whereas with other populations, it's basically minimal. And why is this problematic? Because this all genomic information is being used to build prediction tools to help clinicians to decide who are at risk and also who can respond well to medications. What happens if these clinical tools are not informative in other populations? It could lead to identifying people at risk that are not at risk, or vice versa. And it could lead for people having more side effects of medication that cannot be predicted with these tools. So there's a high need to diversify psychiatry genetic studies. And this is giving you a schematic representation of how it looks across different psychiatry traits. And what color do we mostly see? What color do you mostly see in the screen?
Blue.
Blue. And what blue represents? I know the font is very small. Can you read it?
European.
European, right. So this means that for studies, genetic studies that have looked at ADHD, anorexia nervosa, anxiety, Tourette syndrome, autism, OCD, these are widely and overrepresented by European population, which means that we know very little of what genes are contributing risk in other populations. And I have to say that one of those populations that are very minimally represented are Latin Americans or admixed Americans as represented in this plot. And I'm going to focus on that population from now on. So who are Latin Americans or admixed Americans? Can people name the countries that some of these flags are from?
Puerto Rico, Mexico, Brazil.
Okay, we have Puerto Rico, Mexico, Brazil. What else?
Honduras.
Sorry?
Honduras.
Honduras, muy bien. What else?
Peru.
Very good. One more.
Costa Rica.
Very nice. So I'm sure some of you saw your own flag or your parents' flag or your grandparents' flag or a friend's flag. So that tells you, the extension of like how big these populations are, but also how heterogeneous. What does that mean? How different they are among each other. And this is an illustration in Spanish that we would learn at school in Puerto Rico in our social studies textbooks. And this basically shows the admixture that characterize our Puerto Rican population. And we learned basically that we came from a melting pot of three ancestries: European, indigenous, or Taínos, and African. And I would have to say that in Puerto Rico, we truly embrace our mestizaje, our admixture, feeling proud of our Taino and African ancestors. And we will celebrate the Puerto Rican Day, the Día de la Puertorriqueñidad, in November each year at school, where we dressed up of our ancestry of choice. And I will proudly wear the indigenous one representing a Taina. And we can see also these reflected in current days, with Bad Bunny reaching the top charts with an album entirely in Spanish and proudly celebrating and honoring the salsa music, but also the less known plena and bomba, which are music genres from the criollos and African ancestors that are less known, and now the world got to know it as well. So here I'm showing a world map showing where my DNA comes from. So this shows my ancestral composition. Basically, I sent my sample to a company and they tracked my ancestral background and mapped it based on the similarity of how my DNA looked comparing to DNA of populations from around the world. And you can see that my DNA is highly admixed, from African or indigenous to Europe and Africa, which is basically what the textbooks was telling us. And here I'm showing more data, specific data on admixture. First, in the top panel, I'm showing how you can see different clusters. Each dot is an individual. And you see like more spread clouds and more like compacted clouds. And these are basically ancestral groups like European and African and Amerindian. But here it's mapping on the same map, and you see them in gray here, different populations from across the Americas, from Mexico, Puerto Rico, Barbados, Peru, Brazil. And we basically are all over the place, right? So we are not necessarily clustering to a specific group, but yet there are specific subgroups within the American continent that cluster to each other, but also separated. So this tells you that ancestry is actually a continuum. It's not specific clusters that define a specific population. And that's what we need to represent in our study design when we look at genetics. We need to change every single aspect of a study when it comes to addressing the underrepresentation, and it has to be at every level, from the populations that we study, how the diseases are defined for the study, how we look at the DNA, the tools that we use, and also the analysis and methods that we use to identify these genes associated with disease. And I also have to emphasize that the research workforce, the scientists that do this work, needs to also improve in diversification. These are maps showing where the scientists are coming from that are doing psychiatry genomic studies are mostly US and Europe. But we're slightly changing that pattern. And how do we do that? So in 2019, at one of the conferences where a lot of scientists get together and discuss their science and new ideas comes up and so on, we were able to establish the first Latin America Genomics Consortium. This was together with two other brilliant scientists, Dr. Paula Yussi Rodriguez and Dr. Elizabeth Atkinson. And now from a very small group, from 25 people in that first meeting, we have grown to now, I would say, over 200, representing 10 Latin American countries, the US, and worldwide. I'm updating as I read. With the main goal of advancing psychiatry genomics research in Latin American populations. And on the right, I'm showing how do we do that. So increase the sample, so the number of people that we study, we generate more data that could be added to those studies. We analyze them all together. And also, we develop new methods that can capture the complex admixture that characterizes our population. And also educate and train the next generation of scientists that could do this work locally in their countries. So first, we wanted to take a peek on what psychiatry genetics research looks like for Latin American populations in current days. So we published this paper. If you want to take a look, I'm including the QR code for you to access, where we review the literature, the current literature on this topic. And we basically found that there's very minimal progress and in this field in our Latin American populations. I don't know if you remember this plot from previously, but we were showing you the genetic loci identified for psychiatry diseases in the hundreds. So for Latin American populations, even the best scenario, it's in 25, so not even in the 50s. And this is for very more simple traits like smoking traits. But for more complex traits like even schizophrenia and so on, we're still with no progress at all, or even less than 5. So there's a lot of work to be done across the board. And even in this minimal progress, where is it coming from? Which populations, Latin American populations, are being studied? We see that are mostly coming from the US. So very minimal work in actually countries across the Americas where these Latin American populations are from. So to change that, we decided to work with what we have. So we started thinking what kind of trait, psychiatry traits we can study in genetic studies that haven't really necessarily focused on psychiatry. So we thought about alcohol consumption. How much alcohol do you drink? This is a trait that could be captured in any kind of study. And we did this in a way, not only because it's a relevant trait to study. We know that excessive consumption of alcohol can lead to mental and physical health disorders, but also it's a highly prevalent trait across the Americas, and it's also very highly heritable. And this is basically the study design. I won't go into too much details. What I want you to get at is the multiple levels of analysis that we did to be able to not only boost gene discovery, so discover new genes, genetic variants, but also understand their functionality and test if we can build prediction models that could be performing better in our populations. This is depicting the populations that we study for these analyses. Our intention was to represent not only the Latin Americans based in the US, which we know they're mostly, those are the populations that are mostly studied in current studies, but also populations that are based in other countries. So we were able to collaborate with a big Mexico study. It's a Mexico City prospective study with over 100,000 people from Mexico, and also collaborating with 23andMe, which we know has countries from all over contributing data. We also list the other cohorts that were able to contribute data to the study. And here we're showing, again, the admixture, different clusters that characterizes the ancestral backgrounds of our study cohorts. So initially, we started this study with 55,000, which is very similar to published work, but we wanted more. So we started collaborating with other people, establishing partnerships, and so on. And now we reach close to 500,000 Latin Americans across the world. And we were able to not only capture that peak, basically this is what is called Manhattan plot. We see all the chromosomes here on the x-axis and the p-value. The p-value shows you how important and significant a gene variant is. So the higher it is, the better, stronger the signal and the association with the disease is. So this is what we want to see, like these peaks, but all over. That's why it's called Manhattan, like the Manhattan skyline. So here, initially, with just 55,000, we just see this single peak. When we increase the sample size, we were able to identify more peaks across our genome, but also novel ones that have not been previously discovered in previous studies. And this is comparing from previous studies how our study looks like compared to sample size. So we maximize the sample size, but also in terms of gene discovery, we were able to identify novel variants that have not been previously reported for this population. In total, 14 independent variants were identified, and including a novel variant that have not been previously discovered near the WRN gene, which is a gene that is involved in genome stability and also linked to preventing premature aging. Now, another one of our hits, and this is very interesting, it's a protective variant in the LDH2 gene. It's at RS671. This variant has been identified in previous study in the East Asian population. This is the first study that characterizes this variant in the Latin American population. This variant is important because it's considered a protective variant against developing alcoholism. Why is it a protective variant? We see on the left that people that does not have this variant are able to metabolize alcohol. So ADH1B, the enzyme, metabolizes ethanol to acetaldehyde, and the ALDH2 is able to metabolize it from this molecule to acetate. Sorry. Now, what happens with people that have this genetic variant? Basically, it blocks the action of this enzyme. So it prevents the metabolism of this molecule to acetate. And what happens? What happens is that we started like flushing. Whenever we drink alcohol, we flush, or some people have nausea, some other have tachycardia. And it's considered to be protective because that adverse effects prevents you for keep drinking alcohol. So it reduces the risk. So that's why it's called a protective variant. Now, if people just keep drinking without stopping, without even noticing that it's flushing or it's making you physically sick, then you're at higher risk of developing cancer. So protective variants, because it's like a signal to say to you, please stop drinking, it's not good for you. And this is the first time that we were able to characterize it in the Latin American population. It does not exist it's not prevalent or either European or African. So only East Asian and now Amerindigenous. Here I'm showing that it's highly prevalent in Mexico. We were able, using the data from the Mexico City Prospective Study, we were able to see that in tracts of the genome that are coming from indigenous Mexican, it's 70% prevalent. So very highly prevalent variant. And I'm going to show you a set of slides that I'm going to run over, just give you the take-home message. And it's basically to show the functionality of genes. These help us to identify which genes to prioritize. Could be more significant for alcoholism. These brilliant scientists, and I have to say that these analyses have been performed by my postdoc, Jose Jaime Martínez Magaña, who is from Mexico. These analyses were performed by Paola, but also her postdoc from Colombia, Dr. Luz Porras. They identify how these genes, genetic variants, work in regulating gene expression. My grad student, Melody Rivera Hernandez, was able to identify where in our body these genetic variants have a role in gene regulation. We see that it has a role in the brain, but also in the liver. This is just mapping across the different analyses, the functionality analysis that we run to prioritize which genes are more important than others. And in collaboration with two scientists at Oak Ridge, we were able to, by using artificial intelligence modeling, to not only look at single genes, but also how they talk to each other, the clusters and network that they form and where they belong to. And these help us to know the functionality of that novel hit that we identified in the gene WRN, and to understand better what the functionality of this gene is. Basically, it's working in inflammatory response, and also identify other genes involved in synaptic plasticity and some processes that happen in the brain. Now, for prediction modeling that I told you, like identifying who's at risk, we were analyzing our data that we generated with the genomic study compared to a genomic study that is coming from European population. And I would have to say that we saw it all over the place. Some populations, this is geographical clustering, so looking from countries, how the populations, the risk scores were performing. We saw that for some populations, our model worked better, but some others, the other model that was with European populations worked best. Now, we use also machine learning approaches to cluster our populations into different like subgroups. And that's how we form these different clusters. And we see that, again, variability across the board. Some clusters, our analyses, our prediction model perform better than others. And also one last take is that we were able to look at from the genetic variants that we identified to map from the genome segments where each segment was coming from. Is it coming from the European or Amerindian or African? And we saw that our hits, the ones that we identified, are mostly coming from European and Amerindian tracks. So basically, we proved that studying at mixed populations like the Latin American enhances gene discovery. We look at that, we identify novel functional pathways discovered beyond the alcohol metabolism. And we see a variable prediction of our risk scores, which is driven by the ancestral background composition. In the review paper that I mentioned, we discussed the gaps and challenges, but also proposed recommendations and solutions, and you can access them there in the QR code. These are pictures of the consortium that I thought you might be interested, and fun to learn about our community. This is in each of the conferences that we were able to meet. This is the most recent one. The first time that this Congress is done in Latin America was last year in Cancun, Mexico, which we had a great turnout. This is a list of other consortium focused on Latin American populations that we also publish in our work. Briefly, I'm not only working in the consortium, I'm also part of the All of Us research program, which the idea is to diversify genomic studies across the board in the US. And this is a description of our team here at Yale, our partners with Junta and cultural ambassadors, and our events in the community. I didn't talk about it here, but we also look at across the board, like understanding the functionality of those genes. We also look at neuroscience, and we study postmortem human brain, which is another cool part of my research. Now, a little bit about me. I'll be very brief. As I already mentioned, I'm from Puerto Rico, specifically from a mountain region, a mountain town called Cayey. And previously you saw my sisters, but this is my mother and my father. As mentioned, I did my bachelor's in Universidad de Puerto Rico in Río Piedras. I was able to work at a lab during college, with what basically formed my research career from very early on. Very important that I wanted to highlight the support of my family throughout. And these are different research experience that I had during my college, so highly recommend. From Puerto Rico, I went to Chicago for PhD. This is my PhD thesis committee. Make sure to like people that surrounds you, that it's a strong network and role models. And also showing the different like research papers, leadership roles that I was able to be part of. I met my other half, Jose, during the PhD, and this is a picture of my family attending my graduation. From Chicago, then I went with my husband then in New Haven, where we got married in Puerto Rico, but I started my postdoc in the lab of Joel Ganenter here in the Division of Human Genetics, participated in a bunch of leadership roles, I was awarded multiple fellowships, did publications, but also more importantly, I had my first son, Janitza Montalvo, who is now 7. And last but not least, I started my own lab here at Yale and started off a small group, now a little bit bigger. Was able to get some awards and grants that were able to build my lab as we know it now. Publish papers in high impact journals, and also being able to do some of the leadership roles that I talked to you about in the consortium and so on, and had my second son, Jose Luis, who is now 5. And here the font is very small, but I'm happy to distribute these through email. But I wanted to depict that all those accomplishments that I mentioned, those are the tip of the iceberg. Below it, I'm listing there all the challenges and obstacles that made it very hard to get to where I am. But also in the yellow circles, showing things that helped me to overcome them and get to where we are now. So happy to chat more now about this. I could go more in detail if you want. And yeah, I just want to show my my email for those interested. I can also put it in the chat. This is a QR code of my lab website. I do need to update it though. That's on my pipeline of things to do. And also a picture of my lab, picture of the consortium, and updated picture of my family. And thank you so much. Hello?
Thank you so much, Dr. Montalvo Ortiz. Sorry. Thank you so much, Dr. Montalvo Ortiz. That was incredible. Muchísimas gracias a la doctora. Muy buena presentación. All right, everybody, this is your moment. Este es su momento. So, who has a question? Don't be shy. There's no wrong questions, only curious minds. No tengan miedo. Cualquier pregunta es una buena pregunta. Así que pueden, you can either open your mic and ask a question, or you can write it in the chat. And if you're on YouTube, you can do the same thing. We'll read it for you. Pueden abrir el micrófono o escribirlo por el chat, como quieran. Vamos a leer su pregunta.
How long does it usually take to make a medicine for a disease?
Okay, that's a good question. So it can take a long time. It depends. There's a whole push of working with what we already have. So what that means is that from the medicines that we already have, trying to rediscover if they could also work on other diseases other than the one has been initially targeted for. For drug development efforts, that takes longer, from characterizing the mechanisms of how the disease works, but also medications that target those mechanisms. So that could be genes, proteins that are disrupted in the specific disease. And then for developing the medication itself, that's a whole like different field, like biochemists and so on, that makes sure that the drug targets the molecule or so on that needs to target. But also there's another phase of, testing it in animals, but also testing it in humans where the clinical trials come, which are essential because we don't only want to know if they work, we also want to know if they're safe. And that's a critical part of developing a drug, and that can take very long time. There's ways to try to make things faster. For example, what happened to COVID, leveraging on existing research. How can we prioritize efforts to make a drug faster when there is a real emergency, like global emergency to respond to. So, but if the resources are spread out, I would say, yeah, it could take a long time.
Thank you.
Sure.
Well, we do have a question from YouTube. Someone asked, how do they find the genes? So I'm guessing they're really interested in how you actually locate the genes in the first place.
Okay. Yes. So basically, we sequence the genome of the person. We could use these using platforms that basically they have little probes that mimics the sequence of specific fragments across our genome. And then when it pairs, you identify the exact sequence of those fragments and put them together, and you kind of like try to map out the entire genome. Those platforms are cheaper and more doable for making large-scale studies. Nowadays, people are moving more into whole genome sequencing, which is more expensive, somewhat more affordable than it was 10 years ago now, but yeah, it's still expensive. But what it tells you is like every single nucleotide is read. So you have information not only about common variants, which are variants that are commonly prevalent in the population, but also rare variants. And those could be identified only if you have the entire library of your genome, like all the books that are in the library. And once you sequence them, you do an analysis. So basically, you do what is called linear regression. It's looking at the association of a genetic variant with the disease. So people that have the disease and people that doesn't have the disease, whether the variant is more prevalent in people that have the disease. That's what you want to look for, right? So once you identify that, you map them in that Manhattan plot that I showed, showing each of the dots represented a single nucleotide polymorphism, so a genetic variant. And that's how we know, depending on the significance of that association, we can disentangle those variants that are indeed associated with the disease from those that are not. Now, one thing is association, and one thing is like causality. And that's where the whole functionality of understanding the significance of like the functionality of those genetic variants comes to play. We want to understand if this association is like just a mere association, or is it doing something biological meaningful that is contributing to the disease risk. So that's basically, I went from generating the data, and the DNA could be taken from a blood sample, saliva sample, like in the forensic TV shows that you see, it's just basically the same. And then the technology, I would say, from the platforms or arrays that are used to the sequencing the whole thing, And then the analyses, which is regression models that we use to look at this across the genome, like associations. It's like for each SNP if it's related to people that actually have the disease.
I think Sharif had a question. Sharif, do you want to ask it out loud?
Okay, we'll ask it for Sharif. Sharif asks if there is any proper cure for any type of cancer.
Well, cancer is not necessarily my field, but I would say that I know it's a field that is advancing, and like there's a lot of progress in trying to identify novel medications that are less harmful, but yet also more effective. There's not such a cure for all cancers because each cancer is defined by specific cell type and specific mechanisms. So you need to design medications that are specific to that, cell types that is harmed during cancer or the specific organism that it targets. So, but certainly, yeah, there's a lot of work to be done as well in that area.
Any other questions that you guys have for Janitza? Don't be shy. You had a lot of questions earlier.
You can ask her anything. It can be about her work, but also, like Joanna said, you can ask her just simple questions about school, if she liked it, if it was hard, if she has any advice for students that want to go into science.
Oh, someone has a question.
Go,
Norma,
go.
go ahead.
Puede ser en español también. ¿Sí? Or English, both.
Norma Lucia. Oh, Naomi. Naomi's wondering what inspired you to do your job?
Okay, oh, that's a beautiful question. Okay. So I have to say, my curiosity about learning new things, it started off with an interest in neuroscience, and I talked about this in the other talk in Juntas Podemos, Semillas de Triunfo. My curiosity in neuroscience was through the Alzheimer's disease that my grandparents suffered. So I have two of my grandparents officially were diagnosed with Alzheimer's. I think three of them, or at least one of them more, had dementia at some point. But it was very impactful for me when I was, I think, your age, high school or so on, middle school, around that age, where my grandparent, we started visiting him again, and he's just stopped recognizing us. And he would just look, have that lost look on his face, like he wouldn't recognize my dad or anyone else. So, it just awoke a curiosity in me, like what happens in the brain, what makes a person to stop remembering their family. And then when I started neuroscience research in college, in Maldonado's lab, I studied drug of abuse there. I studied cocaine use disorder, and we studied in a rat model. And if you live in Puerto Rico, and it also happens here, you also see the great impact that it has in a person's life. But I was curious about, from two people with exactly the same background and trajectory, what makes one person more vulnerable to become addicted to cocaine or opioid, fentanyl, and the other person doesn't? Like, what makes them different? And I think that's where I feel like I fall in love not only in neuroscience. Like when I got to PhD, I started learning about genetics and epigenetics, which I didn't touch here. but would love to also talk about, in an email or if you're interested, which is basically what, the experiences that we have throughout, the stress that we have, the trauma that we live, the drugs that we take, how those shapes not only the experiences that we have, but also our biology. So we know that, as I mentioned, our genes does not determine 100% who we are, but also the experience that we have through our lives. And that could be imprinted in our biology. Like that could be whether a gene is turned on or off, and that turns on a whole like domino effect of downstream effects that can have. So there I learned that we are more plastic, dynamic than we initially thought. So it's not that you could develop Alzheimer's or become alcoholic just because you have this gene or genes or genetic variant, but also, the trauma that you live, the stress that increases even further that vulnerability to end up developing the disorder. So I think I would say my curiosity in science started off by trying to understand how the brain works, but it just continuously was shaped with all these new questions that arose as I kept learning about, what other things out there are shaping us. And that's what my initial slide on, what makes us, what shapes our identity. But also, how can we change it? I think that's also powerful to learn as well.
Great question, Naomi. We have another question from Sharif. How many years did it take you to study? In her case, it was science, but it's a science with a medical focus. So how many years did it take you to get where you are, and what was the most difficult part?
Okay. Oh, the most difficult part. Okay. So I started off as a 5 year. I did 5 for bachelor's. I took my time, I was in no hurry. I wanted that experience of like all the research experience that I showed. And then 5 years of PhD after that. So that will be 10. And then another 4 of postdoc, 14. A postdoc, you're considered like already a scientist. So I don't know if I should count that. But yeah, it's a long journey, but a fun one too. Like, I don't know, I saw in the chat, like Javi mentioned, like you get to travel. I was able to, from Cayey, to go to Río Piedras for my bachelor's. Then go to Chicago, and that's how I learned like what snow was, because I've never seen it before. And I told this anecdote when in the other talk, wanted to share it here too. Like the first time I saw snow, like I felt like that was a new trauma for me, because it was like in December, early December. I was on my way for my final test, fundamentals neuroscience, which is a very heavy load course in the PhD, the first year. And I woke up at 6 and I was out of the door. It was 6:30. Had to take a bus, and then I got out, and there were like 2 feet of snow, and I was like with my sneakers on and a fall coat, like not even a coat, not a proper coat, just like the one that you wear before springtime, that kind of coat. So I just had to walk to the bus, and I ended up like all wet and taking the test, like with like, oh my God, shivering of cold. So, but it was fun because like Chicago is a really fun city to be at in the summer. I think it really compensates. It's a beautiful city in the summer, a lot to do. And then the conferences that you get to attend outside, like you got to go to new countries. I've been to like Singapore, I've been to Italy, Germany, Mexico, Peru. So and you get to know other researchers as well, which is really fun to do. People that do science different than you do, like not necessarily neuroscientists or geneticists, you get to know like other people in other disciplines. So, and I have to say, the toughest experience for me, I don't know. In the other talk, I actually talk in each of the slides that I show you the accomplishment in each of the phases of the career. I actually show the challenges and obstacles first. I wanted to talk more about science in this one, like change it a little bit, but I go into detail about in each phase what kind of challenges I had to encounter. And I would say each of the phases have their own unique challenges. But I also have to say that many of these challenges also are recurring. You're going to see them over and over and over, like imposter syndrome, with the work-life balance once you have a baby, how expensive childcare is. You're not thinking about that now, but later on. But also the fact of moving away from your family, learning a new language, and the sense of not belonging that still happens up to now. New challenges now, I would say, being the only women in many, tables that are where decisions are made, being the only Latino background person, and fighting against this stigma that people has of that you're there because you're filling up a spot, like minority spot. That happens. So it's like a constant fight against the society, but also against yourself to try to overcome, those thoughts that could be your worst enemy. So I would say you have challenges throughout. I cannot choose like one that is tougher than the other one. Each one comes with its own monster. No, but it's also you learn how to fight it.
Okay. Yeah, I was about to say one more question and we can wrap up. I think Sharif asked, how many languages do you speak?
Oh, only two. That's it.
English and Spanish.
I wish I know more, but
Janitza, maybe you can just end with some inspirational words or words of encouragement for our students.
right. Absolutely. I think I will have to say I'm going to share the picture that I showed you at the very end with the mountain sort of iceberg. But I have to say, like, try to fight your sense of not being shy, not being like that you're not good enough or that you cannot do it. There's always people that are gonna do that for you. So try to not like that additional person not being you, because the only person that will advocate for you will be yourself. I will have to say also build your own community that believes in yourself, that makes you a better person, and identify those role models, people that look like you, but surprisingly, there are also people that necessarily doesn't look like you that could be strong allies as well, and sponsors. So try to identify those as well. Don't be shy again to ask for help. Be persistent on things that if they don't work at the first time, you just keep going and try to do it again. And believe in yourself and take care of yourself as well.
Okay. Thank you so much, Dr. Montalvo Ortiz, on behalf of everybody here in the Seeds of Success program and everybody watching on YouTube. Thank you for sharing your story, your science, and your time with us tonight. I think I speak for all of us when I say that you are truly an inspiration. And to all of our students, please keep asking questions, keep being curious. You belong in science, and stories like these are proof of that. Todos nuestros estudiantes, por favor, sigan haciendo preguntas, sigan estando curiosos, sigan creyendo en sí mismos, que lo que nos compartió la doctora Montalvo Ortiz les sirva de inspiración para saber que ustedes pueden lograr eso mismo y todo lo que se propongan. Thank you so much, everybody.
Thank you. Thank you, Dr. Montalvo Ortiz. Thank you, Anna. Thank you to all the Seeds of Success students and for everybody who joined us on YouTube.
Thank you, everyone. Gracias a todos.
Gracias.